Can I ask if anyone had trouble logging in either today or in the first lab?
I didn't have any trouble.
I'm not hearing anything. I had real trouble logging in for the first lab. I had to go to www.zoom.com to log in. I wasn't able to log in from the Clark College website. They have a link to Zoom, and it kept telling me unauthorized access. Um, the college has been having trouble with Zoom and Respondus Lockdown Browser. If you remember, I was trying to change something about the quiz and wasn't able to get my changes in. And then also, I noticed the last quiz, I had made the setting so that you would not do the environmental scan before the quiz, that you were supposed to do the environmental scan during question one. Well, the computer or whatever, Respondus did not keep my change. And that is why the last quiz had an environmental scan before the quiz. I told you from now on, you will be doing your scan in question number one. And I have checked for the next two quizzes that my settings have taken. So that if you're going to be doing a environmental scan to get the points for it, you're going to need to do that scan during question one. If you do not do that scan during question one, you will not get the points for that question. And I'm going to increase the points. The next quiz, the question will be worth 2 points. After that, the scan will be worth 3 points. So you have to learn how to do the scan. Some people are still failing to give me a satisfactory scan. And mostly you're doing a scan too high, and so you're scanning to the left and to the right of your face. I need to see where your computer is sitting, and then the scan to the left of where your computer is sitting, and to the scan where the right of the computer is sitting. And I want to see the complete surface, meaning the edge of which you can't see, I know, but the edge of the table or desk or wherever you have the computer sitting. And I have warned people that Respondus says do not have the computer on your lap or on the bed or a soft surface. I'm not sure why they're giving that warning, but I would follow their warning, meaning there must be a reason why they're giving that warning, and I would put your computer or laptop on a hard surface. Any questions about any of that? All right. I'm not showing my screen yet because I wanted to pull up the grade book and state some things I've got graded. I meant to do that during the lab, which is when I normally do it, but because I was late getting into lab one, I totally skipped it. So we're going to go through the grading in the lecture, which is unusual. I've got X credit picture graded. I've got worksheet for the lectures one and two graded. I've not graded lecture three. I'm going to hope to get that done tonight or tomorrow. I have the Plagy quiz and Plagy practice quiz one and quiz 1C graded, at least for question number one. For the plagiarism quiz, I have opened that quiz. If you did not get 17, excuse me, if you did not get 19 out of 19, other than you can miss question one, any other question you miss, you will get a penalty. And the penalty is you'll get 0 on the infectious disease project. So I've opened the plagiar quiz again. Let me see what the date is here. You can take the Plagy quiz again until October 31st. If you haven't used all 7 of your attempts, you can use uh, your 7 attempts. And if you used all your 7 attempts, I gave you one more try. I suggest you do not use a Macintosh computer because everyone who was not able to see the answers was using a Macintosh computer. So either the computer or Canvas or the quiz is not working correctly with a Macintosh computer because you're supposed to be able to see the both your correct answers and the incorrect answers one time after you take the quiz. And like I said, I know it is not working correctly with a Macintosh computer. And if you have a Macintosh computer, you should tell the Canvas people that the quiz was not working correctly on Canvas and you could not see the correct answers. I have put in an error statement, but I'm only one instructor, and it will really be up to a number of students. If a number of students complain about it, they'll actually do something. And I think I sent out an email or announcement on that, and I explained how to put in a request for fixing this problem in Canvas. Anyways, you have at least one more attempt. You might have several attempts until October 31st to get 100% on the Plagy quiz. You can miss the first question, but every other question, if you miss it, you should take the Plagy quiz again. All right. Otherwise, you'll get 0 on the infectious disease project. And last term, for the first time, I actually gave somebody a 0 on the infectious disease project. Any question about any of that? All right, let me go back to the grade book. I have moved over the, meaning I got the environmental quiz and the retake of the environmental quiz graded, and I have moved over the points for the retake of the environmental quiz to the environmental quiz. So if you took the environmental quiz, you got the points of the retake. If you took the retake of the environmental quiz, you got the points of the retake for your environmental quiz. There was an exception, and that is some students, I don't know why they took the retake because they did fine on the environmental quiz. And I just left their score the way it was. I did warn you that you actually would get the points of the retake, but I decided in that case not to uh, not to do the change. And then the retake of the environmental quiz is now not counting in the grade book, so you won't see it in your grades, but you can access the quiz by coming through the link on the first page of our Canvas website. And then worksheet lab 00. And most of worksheet lab 1 has been graded. I think there are a couple of students in worksheet lab 1 I haven't got graded. I'll try and get that graded. Well, not tonight, but maybe tomorrow. Worksheet lab 2 and worksheet lab 3 are graded. For worksheet lab 3, if you didn't get 100%, you can resubmit the worksheet by this Saturday. And I think this Saturday for worksheet lab 2, let me check on that to make certain. I think I got worksheet lab 2 graded so late. Yes, you can make your correction until October 18th for both of those labs. I have not started worksheet lab 3. I hope to get that finished by Wednesday morning, in which case the second lab will be this weekend. But if I do not finish the grading for worksheet lab 3, then it won't be the second grading will not be due this Saturday. It'll be next Saturday. And I'm not using the due date for the second grading. Remember, I'm using the until date for the second grading. And that's because Canvas is not set up to grade an assignment 2 times. Any question about any of that?
I do have a question about the plagiarism quiz.
Uh-huh.
So if I've got like a 19 out of 19, do I have to retake it?
No, you got 19 out of 19. You don't need to retake it.
Okay, thank you.
Somebody answer. Oh, that's a test student. Never mind. I didn't even know you could get into the final A. Somebody put in an answer, but that's the test student that um, it must have carried over from last term. All right. Or actually, this actually carried over from last fall. Any questions about the grading? If not, I'm closing down the grade book. And that's not working right. All right. So let's go to the syllabus. I'll share my screen here. Just a moment. All right. Today is October 14th, and we are supposed to be finishing chapter 3 today. Hopefully, we'll succeed. You do not have a quiz this week. Quiz 2 was moved until week 5, which actually I think on the web page it was correct, and on the quiz it was correct. It was only in the syllabus that was I moved it. Any questions about any of that? All right. Um, I think I'm going to close this down and I'll restart it for the lab. I have that we had talked about the gram stain and went through the procedure and the colors of the cells to this point. Any questions about the gram stain or anything? All right, let's talk about the gram stain in greater detail. First, you always do the gram stain on a bacterial smear. Remember that the smear is not part of the stain. You can make a smear, a bacterial smear, and you cannot stain it. You can use it for another purpose, or you could look at the cells unstained. The gram stain is a stain that always starts with the bacterial smear. Not all stains do that. You could stain cells without making a bacterial smear. And then you have to air dry and heat fix it. That is important because the procedure of the gram stain will wash off cells unless they're stuck to the glass of the slide. And how you stick them is air drying the cells and then heat fixing them to the slide. The first thing you do is you flood the slide with crystal violet. It's the primary stain for 60 seconds. Crystal violet sticks to the peptidoglycan and the bacterial cell wall and will stain all the cells the same color, purple. It's considered the primary stain because it's used first and it stains all of the cells. Any question about any of that? After staining the cells for 60 seconds or 1 minute, you then dump the stain off and then wash the slide gently with cold running water. Now let me state in the gram stain, after each step of the gram stain, you always wash the slide with cold water gently. And you don't need to, if you're giving me the steps of the gram stain, you don't need to tell me flood the stain with crystal violet, then wash with cold water. Flood the stain, the slide with grams iodine, then wash with cold water. You don't have to put in that wash with cold water all the time. When you're giving the procedure, you only need to state it once, and then state, and after each step, we do this step, meaning wash with cold water after each step. Any question about any of that? All right. Because crystal violet sticks to the peptidoglycan and stains all the cells will be staining a bluish purple color. And that's true of the gram-positive cells and the gram-negative cells. After you wash off the crystal violet, and then you get the worst of the water off, you then flood the slide with grams iodine. Iodine is not a stain here; it is a mordant. The grams iodine increases the affinity of the crystal violet for the specimen. What the grams iodine does is it complexes with the crystal violet and makes a larger molecule inside the cell. You apply the mordant or the grams iodine for 60 seconds or 1 minute, and then you discard the grams iodine and then gently wash the slide with cold water. Once again, the color of both gram-positive and gram-negative cells is the same. It will be a purple color as opposed to a blue purple color, meaning you can detect a slight change in the shade of purple, but it is purple. And both the gram-positive and the gram-negative cells staying the same. All right, then you wash the slide with cold water, shake off the worst of the water, and then you apply the decolorizing agent. The decolorizing agent I'm going to discuss is acetone ethanol, and it's a stronger decolorizer than we had, I think, in the last lab where they just used 95% ethanol. The decolorizer, meaning in this case the acetone ethanol, washes the crystal violet iodine complex out of the gram-negative cell walls, but not out of the gram-positive cell walls, as long as the gram stain is done correctly, meaning you do not overwash with the decolorizer. It is considered the differential step of the gram stain because the gram-negative cells will stain a different color than the gram-positive cells. Let me get an example here. So when you're flooding the slide with acetone ethanol, the decolorizing agent, you do it for about 5 seconds. But a better way to do it is to apply the decolorizing agent. Hopefully you can see this. Pretend this is the slide, pretend this is the dropper having the decolorizing agent. You put the decolorizing agent on and just move it back and forth across the slide. So the decolorizing agent is running down the slide. Once the blue stops streaking on the slide, you stop adding the decolorizing agent. And that will happen in about 5 seconds. That's the best way to do it because you're not putting on too much decolorizer, because once let's start with the other way. If you're putting on too little decolorizer, the decolorizer will still be running blue, streaking blue, running down the slide. And so you want to add the decolorizing agent until the blue stops running. Once the blue stops running, you should stop adding the decolorizer, because if you keep adding more decolorizer, given enough time, and we're talking about seconds here, the decolorizer will pull out the crystal violet iodine complex from the gram-positive cells, meaning you are over decolorizing the cells. So you apply the decolorizing agent for about 5 seconds, and the best way to do it is to simply stop adding it when the blue stops streaking on the slide. And then the gram-positive cells will stain purple, and the gram-negative cells will stain clear, meaning the ultraviolet iodine has washed out of them. You need to wash this slide immediately. The reason is that even if you dump the decolorizing agent off the slide, there will be trace decolorizing agent on the gram-positive cells. And given enough time, that decolorizing agent can pull the crystal violet iodine complex out of the gram-positive cells. So you need to wash immediately with cool water. And you gently wash. So the color of the gram-positive cells will be the purple color. The color of the gram-negative cells will be colorless after the decolorizing agent is applied. Any question about any of that? And that's why it's called the differential steps, when the gram-negative cells differ from the gram-positive cells. You then dump off the worst of the water and flood the slide with safranin. Safranin is called the counter stain because it's the second stain used, and it's a different color than the primary stain. You can flood the safranin for 1 to 2 minutes, and then you dump the excess safranin off, and then wash the slide gently with cold running water. So in my procedure that I give you, if you simply state that you stain the cells and apply the agent always for 1 minute, you will be correct, with the exception of the decolorizing agent, which you should apply for only 5 seconds, at least if it's acetone ethanol or if it's acid ethanol. The only exception is if it's 95% ethanol, then you apply the decolorizing agent for a longer period of time. And once again, the best way of doing that is simply to add the decolorizing agent and wait until the blue streaking stops. If it's 95% ethanol, you'll just be using more ethanol and it'll take longer, something like 20 to 30 seconds. The blue will stop streaking if you're using 95% ethanol. All right. Now the safranin is a pink dye, and it does stain all the bacteria cells. But after the safranin stain, the color of the gram-positive cells is purple, and the color of the gram-negative cells is pink. Any questions about that? You then simply blot the slide dry. Let me get some paper. A blotting paper is closer to like a thin cardboard, but I don't have blotting paper with me, so I'm just going to use this napkin. And what you do is you rip off the blotting paper from its paper mound. It's a wad of paper. And then take your slide and put it in the fold, and then just press the blotting paper down on the slide. The purpose is simply to remove enough of the water so you can view the slide under the microscope. And it does remove enough of the water so that you can even use oil on the slide. Do not take the blotting paper and move it on the slide because that'll pull off cells. Do not move the slides against the blotting paper because that'll move off. You just want to, I don't know, pat it down to remove the most of the water. Any question about any of that? And then after doing that, you can examine the slide directly under the microscope. There isn't enough water to hurt you. All right, so the basis of the gram stain. The gram-positive cells are purple after the procedure, and the gram-negative cells are pink after the procedure. Why? What's the basis of the differential staining? It has to do with the differences of the cell wall. The gram-positive bacteria have a thick peptidoglycan layer, and the Graham's iodine crystalloviolate complex gets stuck in that peptidoglycan layer. The gram-negative bacteria, on the other hand, have a thinner peptidoglycan layer, and the crystalloviolate iodine complex washes out of that thinner peptidoglycan layer with the decolorizing step, assuming the decolorizing agent is applied for the correct amount of time. Any question about any of that? All right, let's go back to this one here. Safranin is a pink dye, but I stated right there, safranin gets in all of the bacteria. So why aren't the gram-positive cells either purple and pink or a purple-pink color or even pink color because you're applying the pink after you apply the purple? Anyone have a guess? I don't expect anyone to know, but I'm just asking.
Is it because the peptidoglycan layer is so thick it doesn't wash out?
No, we don't wash the safranin out of the cells. All you do is you apply the safranin, and then you do uh, wash the cells afterwards, but it didn't remove the safranin from the gram-negative cells. So it tells you it doesn't remove it from the gram-positive cells as well. The reason why the cells are purple is simply because purple is a more intense color, and our human eyes only sees the purple. But if we were to look at a cell, let's say this is a cell right here, you would see under the electron microscope regions of the cell which stain purple and regions of the cell which stain pink. So you can see the pink stain if you look at it under the microscope and then you blow up the slide greatly. And I think most of the slide actually doesn't have a color, but the purple is what our human eye focuses in on. So when we look at the gram-positive cells, they only stain purple. But the pink dye is actually in those cells. Okay? You just can't see it, at least with the human eye. And I don't think you can see it under the light microscope either. To see it, you have to look under with electron microscope. All right, let's take a look at the difference between the gram-positive cell and the gram-negative cell. Let me blow this slide up. The gram-positive cell has a very thin lipid bilayer that we call the cell membrane or the plasma membrane, and then a very thick purple layer, which is the thick peptidoglycan layer. This is showing 3 layers of peptidoglycan. That is a mistake because they're not going to draw a whole bunch of peptidoglycan layers. The gram-positive cell is at least 30 layers of peptidoglycan, and it may be hundreds of layers of peptidoglycan. One layer on top of the other, meaning this layer is the bottom one, and then I think that layer is the next one, and then that one's the next one above it. So just layer upon layer of peptidoglycan. And it's a gram-positive cell. There are some other molecules in the gram-positive cell. Let me go ahead and talk about that. Nope, I'm not going to talk about it now. I'll talk about it in a future lesson. There are some other molecules, but the main molecule in the gram-positive cell, by far, is peptidoglycan in the cell wall. The gram-negative cell differs, and it does have the same lipid bilayer that we call the plasma membrane. It does have a thinner peptidoglycan layer, assuming the bacteria has a cell wall. It has a thin peptidoglycan layer. But outside of that peptidoglycan layer, there is another lipid bilayer. And that lipid bilayer is called the outer membrane of the cell wall. So the outer membrane of the cell wall is part of the cell wall, and the peptidoglycan layer is part of the cell wall as well. That outer membrane is a bilayer similar to the plasma membrane. There are some other molecules in it. But that layer there of the outer membrane of the cell wall is the same as the bilayer of the cell membrane, meaning the main molecule is a phospholipid molecule shown there. It's hard to show, but you can see the head and the tails. Let me blow that way up. There, that's the phospholipid right there, the tail and the head. So this layer is mostly phospholipid, but there are other molecules in there. There's proteins spanning the membrane. There can be carbohydrates. There can be other lipids. Now, is that true for bacteria? Animal cells have cholesterol, which is a lipid in the cell membrane, but bacteria do not, unless they don't have a cell wall, in which case they have another sterol other than cholesterol, but it is a sterol, similar to cholesterol. Anyways, in this layer of the outer membrane of the cell wall, you'll note that it has other molecules in it, namely this lipopolysaccharide, which has a fat blue lipid there, which is called lipid A, and attached to lipid A is an O polysaccharide, and that's sort of these hairs going up. And together, the lipid A and the O polysaccharide make the lipopolysaccharide. Lipid A is important because if the cell wall is a gram-negative cell, meaning it has a cell wall, then it has lipid A, and lipid A is an endotoxin. If you do not know, a toxin is something that is toxic to us, and so any patient who gets a gram-negative infection, as long as that species has a cell wall, it will contain lipid A, the endotoxin. Any question about any of that? And then there is another protein we find in the gram-negative cell that you do not find in the gram-positive cell. It's called the porin protein. The porin protein is a channel protein to allow molecules to enter and leave the outer membrane of the cell wall. We do not have the porin protein in the cell membrane. What we have in the cell membrane is a transport protein, which each transport protein pumps essentially one and only one molecule across the cell membrane. For example, a cell that wants fructose will have a transport protein to bring the fructose into the cell membrane, through the cell membrane into the cell. And it has a separate transport protein to bring the sugar glucose into the cell membrane and into the cell. There are not transport proteins in the outer membrane, only in the cell membrane. For the glucose and the fructose, they have to come through the porin protein. And this porin protein is a channel protein for many molecules to enter or to leave the outer membrane of the cell wall. Any question about any of that? Let me make sure I've talked about everything I want to talk about. All right, there are some molecules in the cell wall, either in the outer membrane of a gram-negative cell wall or in the peptidoglycan, including lipoproteins, and other carbohydrates, and obviously proteins. Like there's a protein right there going through both the cell wall and the outer membrane. And there's another protein there, which I've already named the porin protein. Any question about any of that? Oh, come on. So the crystal violet iodine complex forms in all cells, both gram-positive and gram-negative, but the crystal violet iodine complex is larger than crystal violet iodine alone. During the alcohol acetone wash, the gram-negative cells have the crystal violet iodine wash out of the cells because peptidoglycan layer is thin, and then the lipopolysaccharide layer, meaning the outer membrane of the cell wall, that outer membrane will be destroyed by the ethanol in the decolorizing agent. Phospholipids are destroyed, as well as the other lipids are destroyed by the ethanol as well as the acetone. And so the outer membrane will disintegrate. That will allow the crystal violet iodine complex to wash out of the cells, and then the cells will be colorless after the acetone alcohol wash. The gram-positive cells, the crystal violet iodine complex, cannot wash out of a thick peptidoglycan layer. The cells will therefore remain being purple in color. Any question about any of this? All right, here's a picture of a gram stain, a gram positive rod, a gram positive cockeye. It's not labeled there. Here we have a gram negative vibrio. Remember, that's a curved rod or a half a spiral. I'm looking for a gram. There's a gram negative rod. And then there is a gram gram-negative spirilla, which isn't labeled, but there's one there. Any question about any of this? All right. When you do the gram stain, you may see something like this. If you were to really come into a microbiology lab and stain the cells with a gram stain, you may see something like this. How do you read this slide? You read the slide from areas of the stain where the stain is easy to distinguish and the cells are individual. You do not read the slide in the bad areas where either there's clumps of cells, or in this case, that's actually not a clump of cells, that is a clump of the crystal violet. So that's actually a crystal of the dye crystal violet. And you'll note, let me blow this up, that nearer to the crystal of crystal violet, the cells stain darker than they do further away. Or even there, it's not as dark as there. And what's happening is, I think, crystal violet is coming out of the crystal and then coloring the nearby cells darker. So you want to avoid this dark region because in this region, the cells could be looking gram positive, and that's because of all the crystal violet coming out of this crystal of crystal violet and then attaching to the cells. And then in other regions of the slide, the cells could be pinkish, which this isn't quite pinkish, but it's much lighter than the purple here. So you always read the slide in a good area of the slide. You avoid the bad areas where there's either a large clump or actually a clump of crystal violet. Any question about any of that?
I have a question.
Uh-huh.
If majority of it is bad region, do you have to do it all over again? Or do you, let's say like there's only a couple, do you just read the like one or two that you can see? Or do you just
Talking
do it
about
over again?
one or two cells, or you're talking about one or two regions? Very
Like
good.
like one or two, I guess for both, I kind of just want to get
If it's
understand.
one or two regions, that's good enough. If it's only one or two cells, I would do it over again because you only have one or two good cells.
Oh, okay. Okay, thank
Okay.
you.
All right. All right. Some miscellaneous stuff about the gram stain. You get the most consistent results when the gram stain is performed on young growing bacteria. In the gram stain, you should only be staining cells that are 2 days old or less. If the cells are older than 2 days, the gram stain may not stain the cells correctly. And the main reason for that is if the cells are more than 2 days old, you will have dead and dying cells. And a dead cell certainly may not stain correctly because it doesn't have an intact or may not have an intact cell membrane or cell wall, or both. So when you're doing the gram stain, you should only do it on cells that are 2 days old or less. And if your cells are more older than that, then you should streak out your cells for a day or two and then do the gram stain on the freshly grown cells. And then the gram stain is not universally applicable. There are some bacterial cells which stain poorly, if at all. It's mostly the mycobacteria that stain poorly, and it's because they have a bunch of mycolic acid in the cell wall. Mycolic acid does not allow water-soluble molecules to cross the cell wall, at least not directly. And so the mycobacterium do not take up either water or the dye which is dissolved in water. In my experience, the mycobacterium is always staying gram-positively, but they stain lighter than most other gram-positive cells, and that's because they are resisting taking up the stain. And I've always used cells that are 2 days old or less. If you use cells that are older, then the mycobacteria may not stain gram-positively. They may be gram-variable, meaning some cells will stain gram-positively and some cells can stain gram-negative, even though they are cells of the same species. So note that the results of the gram stain are not always as clear-cut as theory would suggest. If you were to actually do the real unknown project, you'd have to deal with this, I don't know, problem or concern about the gram stain because you're actually doing the gram stain. And the question is that gram stain correct? The main thing is if you stain cells older than 2 days old, you will likely get a gram variable result, and the stain may not work correctly, which I guess the gram negative would always be the case. It's not staining correctly because mycobacterium are gram positive. All right, the structure of the gram-positive cells not only affects the staining, like in the gram stain, it also affects how the cells respond to certain antibiotics, like the gram-positive bacteria. They tend to be killed easily by penicillin G, as well as cephalosporin, another antibiotic, and then many detergents. However, the gram-negative bacteria are more resistant to penicillin G, cephalosporin, and many detergents. The reason why the gram-negative bacteria are resistant to at least these antibiotics and many detergents is because the gram-negative cell has a lipopolysaccharide layer as the outermost region of the outer membrane of the cell wall. And that lipid bilayer of the cell wall, or the outer membrane of the cell wall, is preventing the molecules from entering the cell, and then for the antibiotics, preventing the damage to the cell. And that's why if you give penicillin G to a bacterial infection that is gram-negative, penicillin G is not likely to work. Any question about any of that? Now, when I stated that the gram-negative bacteria are more resistant to these antimicrobials, realize that we do have antimicrobials that do work on gram-negative bacteria, like streptomycin is a lipid antibiotic, and it will go right through the cell wall and the cell membrane of a gram-negative bacteria. Ampicillin and carboxycillin are members of the penicillin family, which work on both gram-negative and on gram-positive. And it's kind of important because ampicillin was actually the third antibiotic discovered, the third member of the penicillin family to be discovered, the first being penicillin G. Any question about any of that? All right. Let's briefly talk about special stains. You're not going to be examined on this, but you may need to do a special stain for your unknown project. So I want to talk a little bit about the capsule stain. This is a stain used for staining for the capsules around a cell. Some of you may need to do this stain for your unknown project. The capsule stain, also called the negative stain, stains both the background of the cell, a lightish purple, and then it also stains the cells a dark color here. And this is probably two stains in this capsule stain, meaning we're probably using a different color or a different stain to stain the cell, and then a different color or stain to dye the background. When you use this stain, the capsule stain, you can easily see capsules around the cell in a cell membrane. And there's a few cells which are only showing the capsule. I'm not sure if the cell is at the bottom of this and out of focus, or if it has been separated from this cell capsule, meaning this cell capsule has been separated from the cell that it belonged to. All right, any question about the capsule stain? Here's another capsule stain. This one is using India ink, which is a true negative stain. The India ink stains the background very dark gray. And India ink also attaches to the cell, but less strongly, and it is staining the cell a light gray. And in between the light gray of the cell and then the background of the slide showing a kind of a blackish color, we see the cell membrane as a clear region including the cell's capsule. And that's how we can detect the capsule of a cell. All right, any question about any of these? If not, that's it for chapter 3. Let's get started with chapter 4 because I want to try and work ahead a little bit. And I promise you'll get out early today. Any questions about anything on chapter 3? If not, let me go back to where I was. No, I'm not finding the 2 here. Oh, here it is. I think I erased it, but let's check. I did not. This must be from previous terms. All right, now let me save it and get out of here. Chapter 4 is the functional anatomy of prokaryote and eukaryotic cells. Even though I'm saying it's about the anatomy of eukaryotic cells, I almost never talk about the anatomy of eukaryotic cells. And that's because you should have had that in your preliminary biology class. And so I do not spend time going over that in the microbiology. It should be just review to you, talking about the different cell structures found within a eukaryotic cell. We will talk about the functional anatomy of prokaryotic cells. I will also talk about the endosymbiotic theory. I'm not seeing that in here. I will talk about the endosymbiotic theory, which is a theory of how animal cells got the mitochondria and plant cells got the mitochondria and the chloroplast. Anyways, here's the major goals and a rough outline of chapter 4. Know the terms. What's the coccus? What's the bacillus? What's the sporella? What's the like streptococcus? What's the staphylococcus? What's the diplococcus? And you could talk about streptobacilli, diplobacilli. What's a tetrad? What's a sarxinia? What is a vibrio? What is a spirochete? What is the glycocalyx? What is the flagella? What is an axial filament? What are fimbriae? What are pili? What's peptidoglycan? What's an endotoxin? What is an exotoxin? What is selective permeability? What is diffusion? What is facilitated diffusion? What is osmosis? What is active transport? What is tonicity? What is a plasmid? What are inclusions? And then lastly, what are endospores? Well, excuse me, not lastly. Second to the last thing, what are endospores? And then lastly, the endosymbiotic theory. And I will discuss this theory and about the only thing I will discuss of eukaryote cells. Any question about any of that? All right, you should also understand how gram-positive and gram-negative cell walls differ. And 3, describe the importance of taxis in bacteria. So this slide is showing you prokaryotes on the left and eukaryotes on the right. Remember, with a prokaryote, we have two domains, domain archaea and domain bacteria. Prokaryote is Greek word for pre-nucleus. Literally, it means pre-kernel. The kernel is used for nucleus, so we're going to say pre-nucleus. Eukaryotes, the word literally is Greek for true nucleus or true kernel. So U means true, karyotes kernel. The prokaryotes have one circular chromosome. That chromosome is not in a nucleus. The eukaryotes have linear chromosomes, usually many, by the way, and these chromosomes are inside the nucleus. For bacteria, their DNA lacks the histone protein, and in eukaryotes, the DNA is associated with histone proteins. In fact, the DNA wraps twice around a big histone protein. The DNA actually wraps around it twice. So the histones are important for DNA wrapping as well as DNA moving. Prokaryotes have no organelles. By organelle, I mean snow-bound organelle. So prokaryotes, no organelles, but eukaryotes do have organelles, at least for the eukaryote parts. And by organelle, remember I'm referring to a membrane-bound organelle. The non-membrane-bound organelle I usually call a cell structure. A non-membrane-bound organelle would be something like the flagellum or an endotoxin. All right, peptidoglycan is found in the cell walls of bacteria only. It is not found in the archaea or in eukaryote cells. And then lastly, prokaryotes divide by binary fission, at least most prokaryotes. What happens is the cell, it may grow, and then it literally splits into two, one half being one daughter cell, the other half being the other daughter cell. And the daughter cells are clones of each other, meaning they should have the same DNA as well as the same molecules that were found in the initial parental cell. Eukaryotes do not divide by binary fission, they divide by mitosis, where one cell undergoes cellular division to get two cells. The third what was I talking about? Sorry, just drew a blank here. Can somebody tell me what the heck I was talking about?
You were talking about how prokaryotes divide by binary fission, and then
Yeah,
eukaryotes by
by
mitosis.
mitosis. And that's about it, actually. Eukaryotes divide by mitosis. They do not divide by binary fission. And prokaryotes, especially the bacteria, most of them divide by binary fission. All right. We're going to talk about the prokaryote cell shape and the cellular growth arrangement, also called the cellular arrangement. We'll then talk about the cell structure of prokaryotes. And then lastly, we'll talk about the movement of material across membranes, particularly across the cell membrane. For eukaryotes, we're really not going to talk about the cell structures. I never do. You guys can review my slides. You can do the reading. if you need support, but I'm not going to lecture on it. I will, however, talk about the endosymbiotic theory, and that is how eukaryotes, it's thought, came to have a mitochondria and a chloroplast. So we will talk about that, but that'll be the end of this lesson. All right, here's some prokaryote cells with their average size. The average really for a prokaryote is between 1 and 2 micrometers. The 10 micrometers, that's getting up to the biggest prokaryote cell, which actually the biggest is, let me think about that. I don't know the biggest. The smallest is about, at least the smallest viable is about 400 micrometers or no, it's not a microgram. It's 0.4 micrometers is the smallest viable prokaryote cell. You can find prokaryote cells down to 0.2 micrometers, but it's thought that those real tiny prokaryote cells are probably not viable. All right, any question about the average prokaryote size? And I need to look up the largest prokaryote cell is. I think it's a little bit larger, not much, much larger than 10 micrometers. And I think that was actually discovered very recently, the largest prokaryote cell. Let me go ahead and just do a search here because I do not remember. I'll just limit it to bacteria. Oops. All right, there's actually a giant bacteria, which is 1 centimeter long. But that's the unusual one. And that's one found very recently. Why the heck is it so long? This bacteria is actually visible to the unaided eye. Of course, a centimeter we can easily see. We can see 1/10 of a centimeter, and even smaller than that. All right, you don't need to know that, but that's the very largest bacteria. And I have no idea why the bacteria is that large. But most bacteria are 1 to 2 micrometers in size. The wrong one. Trying to put a note in here. Okay. All right, this slide is also showing you the different shapes of prokaryotic cells. Most prokaryotic cells have one of three shapes. They can be a coccus, and that's a round ball-like cell. They can be a bacillus, and that's a rod cell. You should note that the rods can have rounded edges like this one, but they can be pointed edges or they can be flat edges. And then the third shape is a spirilla, a spiral like that. Every bacteria, well, I shouldn't say every, most bacteria is one of these three shapes, and the exceptions are some combination of this shape. Like you can have a cuccobacillus, which is an elongated cockeye, or a rod that you've shrunk and shrunk and shrunk, like this one here. If it has rounded edges, you'll eventually get it very close in shape to a coccus, and that we call a cuccobacillus, which I think I've got a picture of that down here. Yeah, right there. This is actually in the wrong location. I don't know why the author put it on this slide, because this is a cell shape, cuccobacillus. Let me blow that up so you can see it better. This is a cell shape. It is not a cell growth arrangement or cell arrangement. And what you do is, like this one here, well, both of those almost look like a rod. That one there almost looks like a cockeye. And what they do is you just shorten them, and then you have something in between a cockeye and in between a bacilli. And that we call a cuccobacillus. Any question about any of that? Now, where were we? I think we were here. You can have three different types of spirals. You can have a vibrio, which is half of a spirillum. So that's the S. You take half of that, you have a vibrio, but you can also call a vibrio a bent rod. If you simply bend that rod, you get a vibrio. And either a vibrio is either a bent rod or half a spirillum. I don't care which you use. Spirillum, the normal spiral, and then you can have a spirochete, and that's a spirillum with multiple bends. There are some differences between these two cells. The spirillum is rigid, a rigid cell. The cell wall and cell membrane is, I don't know, stronger and more rigid than the spirochete. The spirochete is a flexible cell. It can even bend this way and that way. And I'll show you that in a movie, in some future movie in the class, where the spirochete, to change, it moves this way or that way. And to change going north and south, what it can do is bend itself up and then straighten out this way. And now instead of facing east and west, it's facing north and south. The spirillum is more rigid, it can't move like that. Prokaryotes can be some unusual shapes. For example, the Stella bacteria, that's a family of bacteria, actually is a star shape. And that's an actual electron microscopic image of the Stella bacteria. That's the artist's rendition of the Stella. And then there's the Haloarcula, archaea. It's a member of the Archaebacterium, which actually it's rectangular, but they're saying square-like here. It's sort of a square, but that's still a rectangle. There are very few bacteria that are unusual like this, but there are some which do not follow the three basic patterns of a cockeye, bacilli, or a spiral. Most bacteria species are monomorphic, meaning they all have the same shape. Like the Haloarcula, all of them are squarish or rectangular. The Stella, all are star-like in shape. The Staphylococci, all of them are cocci. So they're monomorphic. They have a single shape. But a few bacterial species, the cells are pleomorphic. Even within one species, the cells can have many different shapes for that one species. Most of the pleomorphic bacterial species are bacteria that lack a cell wall. The cell wall tends to give shape to the cell, why the halo are cula, are squarish or rectangular because of the cell wall. And the staphylococci, the reason why they're cocci is because of the cell wall. These bacteria probably don't have a cell wall, and then they take on many different shapes. I don't know even what that shape is. There's like two of them. They're sort of a bent rod, although it's a little thicker on the ends. That's, well, that one there is like a cocci. That one's approaching a cocci, a coccobacillus. Now that one's like a pyramid with a bottom to the pyramid. Pleomorphic. I think I'll end the class here and we'll talk about the bacteria cell arrangements in our next lesson. Any question about anything? All right. Oop, it's not going to show. Well, I will see you at 6:30 if you're in the second lab. Otherwise, I'll see you in the next class. Bye.